गुड मॉर्निंग फ्रेंड्स। आज हम बात करेंगे जेनेटिक्स के एक दूसरे चैप्टर के बारे में जिसमें हम मल्टीपल एलेलिज्म और मल्टीपल एलील्स के बारे में बात करेंगे। मल्टीपल एलील्स एक्चुअली मेंडेलियन जेनेटिक्स का एक एक्सटेंशन माना जाता है, क्योंकि मेंडल ने हमें ये बताया था कि एक जीन के दो वेरिएंट होते हैं, दो एलील होते हैं जो कि दोनों होमोलोगस क्रोमोसोम पर रहते हैं, और डिप्लॉइड ऑर्गेनिज्म में दो ही होमोलोगस क्रोमोसोम हो सकते हैं। ये मल्टीपल एलील्स कहाँ हो सकते हैं, कैसे हो सकते हैं, इनके बारे में हम बात करेंगे, जो कि आपके सिलेबस में ABO ब्लड टाइप्स, उसकी इनहेरिटेंस और उसकी बायोकेमिकल जेनेटिक्स से संबंधित आपके चैप्टर में है। लेट्स बिगिन विद द डिस्कशन। फर्स्टली, we know very well that an individual, a diploid individual especially, have only two maximum copies of a gene, and these two, if are governing the contrasting form of character, for example long and dwarf, tall and dwarf, okay, then these two different forms of a gene are called alleles. So usually majority of genes are provided with two alleles. One is located in one partner of homologous chromosome that comes from mother, and another is located on the other homologous chromosome coming from the father. So that's the reality. In any situation, an individual cannot have more than two alleles if we are talking about the diploid organism. Okay. Then what's the multiple alleles, where they do exist? For example, we can take a rough example. Say one individual is having two variations, okay, A1 and A2. In the same population of that individual or that species, the another individual is having some sort of mutation in the A2, and that's why that A2 is converted into A3. So that individual is having A1 and A3. In another individual, we can think of that he or she may possess the A4 and A2. So if we are concerned with the same gene, that gene is having in that population four different forms. Those are A1, A2, A3 and A4. That means if we are going to study the population, then we will find the four variants of that gene, and these variants are called alleles. That means if more than two alleles, if we want to study, we have to study the population, not a single individual. So that's the overall concept of the multiple allelism. So by definition, or by the simple words, we can think of that not all the genes have the two forms as mentioned by the Mendel. After that, there have been many studies, and those studies proven that many of the genes have more than two forms, and these are called multiple alleles. And this phenomena is called multiple allelism. And since a diploid individual can have only two alleles, one on each homologous chromosome, only two maximum possible alleles he or she can have, no matter how many alleles for the gene exist in this multiple allelic series. So in a very simple way, multiple alleles are basically represented in a population, not by a single individual. There have been many cases of the multiple alleles. For example, the genes responsible for the coat coloration of rabbit, that is the fur coloration, we can consider, that is basically determined by a gene which is having four variants: albino, Himalayan, chinchilla and wild type. Here we can see in this diagram, albino means pure white, chinchilla means majority is white, Himalayan is majority is white like the ice, and chinchilla is more or less mixture of albino and dark shade, that is the black, that means almost a gray, and finally the wild type that is having brown or the grayish coloration. So here we can see there is a gradient in the expression of all these alleles. Here we cannot consider the extreme situation like tall and dwarf. This is tall, this is dwarf. This cannot be the situation. Here the pigmentation is increasing from the albino to Himalayan to chinchilla to wild type. So there is a gradient in the domination of the alleles. That means the most dominant allele is wild type allele that has been designated as C+. And the chinchilla is dominant to Himalayan and albino. And Himalayan is also dominant to chinchilla. So the important character comes for the multiple alleles is there is a gradation of dominance. There is no absolute dominance case. Similar kind of example we will discuss in detail that has been mentioned in your syllabus, and that is the human ABO blood types. Okay. The another example that has been studied in case of plants is the self-incompatibility gene, S gene. And that S gene basically determine or encode ribonucleases, and that ribonucleases will decide which pollen grain would germinate on the style or stigma or not. So that is very important for the reproductive fitness in plants and for the variation in plants as well. That would determine whether the plant would undergo self-pollination or the cross-pollination. And one more example in the classical case of Drosophila is white eye gene that is having more than 100 variants. So one by one we are going to consider all those things. Firstly, we can think of that what is the multiple alleles, that means the same locus, same gene that can have the different kind of mutation. Say in A1 mutation is occurring at this place, but in A2 mutation is occurring at another place. So in those variations, what happens? The nucleotide sequence is changed. Because of that, codon sequence is changed in the mRNA, and finally they would determine the different types of polypeptides having the different amino acid composition. That means all these alleles represent the same locus, but they determine or they encode the different proteins. So the different proteins would reflect in different phenotypes. That's why the situation like this, chinchilla, wild type, Himalayan, we can see in the phenotype. Okay. Similarly, there is a case of Drosophila that we have already said, we have discussed that more than 100 mutant alleles have been recognized for the eye coloration. For example, few of them are the white-eyed variant has been designated as W, the wild type, that is brick red, that is W+, and the third allele for the same gene that we can consider WE, that produce eosine orange color of eyes. So again there is a gradation in the dominance. We have just picked the 3 alleles out of the 100 for the sake of convenience. Now the very important topic coming in our syllabus is ABO blood types. The ABO blood types have been discovered basically by Landsteiner, and for that he received the Nobel Prize. What is the utility of the discovery of the human ABO type blood groups? The utility is this discovery basically determines that the blood transfusion should occur in proper way so that no human loss occurs for the sake of blood transfusion. That means the proper type of blood is given to the proper type of individual. So he told us that there are 4 basic blood types here. That means these are 4 phenotypes, and these phenotypes are, we can say, A, B type, AB type and O type. He told us, he found that these are basically governed by three different types of alleles, IA, IB and that small I alleles. The IA is responsible for generation of A antigen on the RBC surface, while IB encodes B antigen on the RBC surface. Antigens are nothing but a kind of molecule that are immunogenic. That means they trigger the production of antibodies in our body. So this is very important thing for the reaction. What happens if an individual carrying the A antigen is provided with the B blood group? Then the B blood group that contain the A antibody would react to the A antigen, and that would be known as agglutination reaction, and the person would die of blood clump formation. So this is very important for the blood transfusion studies. That's why Karl Landsteiner have been honored with the Nobel Prize. This blood typing is also used for the disputed parentage at the earlier stages. Nowadays it is basically carried by DNA testing or the DNA fingerprinting, etc., etc. But in the earlier studies it was having the initial clue for the parentage. For example, an individual having the IB, that means AB blood group, could not have a parent with the O type, pure O type, because one is IA is contributing to the one parent and IB is contributing to the other parent, and if the parent is O, any of the parent is not having IA or IB allele. So such kind of initial analysis can be done by the blood typing. Now these four phenotypes have been specified by 6 different genotypes, in which the IA and IB, these are dominant to small I allele. So the small I allele combination would determine the O blood group, while the IA or I small I, that would determine the A blood group. And when these IA and IB comes together, they specify a different kind of phenotype, AB blood type. So one by one we are going to study all those things. In simple manner, what we can think, that the small I allele is recessive to IA and IB, and importantly both IA and IB individuals in IA and IB individuals, the IA and IB alleles are codominant because they are expressing simultaneously. So here the principle of dominance proposed by the Mendel is not working, rather than here the principle of codominance occurring. So what happens during the inheritance, we can think of, for example, an individual is having O blood type, then the possible genotypes of his or her parents may be like these options A, B, C, D, E, F that we can see easily. And I insist that all of you students should consider this particular example as an exercise, and then you can match these one so that you can get the better confidence in this topic. Now, the total number of possible genotypes can be determined by this formula, where the N is number of alleles. For example, there are 3 alleles. That means the N is 3. That means 3 and 4, 12 divided by 2. That means 12 divided by 2, 6. So here we can consider again in this same example that there are 3 alleles and the number of genotypes are 6. Similarly, we can find out the number of homozygotes and number of heterozygotes. The number of homozygotes are equals to the number of alleles, and number of heterozygotes, there is a formula. Anyway, and again I would insist, the student community to use these formula in practice, so that again they can have the good idea of these topics or the numerical sections of this topic. Now we are going to consider that we have already discussed that the cellular antigens, say antigen A, antigen B, etc., these are very important in the blood transfusion, as we have already discussed, but still we can continue with that one. If the recipient is having antibodies for the corresponding antigen of the donor, then there would be the agglutination reaction, and this is not called a good blood transfusion. That is a deadly combination. So the antigen must not have the corresponding antibodies. The different kind of antibodies may have. Okay. So in this particular example, we can think of, say, if this is the recipient, O is the recipient, that means it is having the antibody A and antibody B. So any donor which is having antigen A or antibody A, they cannot be useful for that. This O individual can be given only O blood group. That's it. If it is given to A blood group, then the antibody of O individual would react this one, and that would be agglutination reaction. Similarly, we can think of AB blood group. It is having no antibodies, so any kind of blood type can be transfused to them. That's why O blood group are considered universal donor, while the AB blood group are considered as a universal acceptor individuals. Similarly, A individual can be given only A blood group or the O blood group, and B can be given O blood group and B blood group. So that was the situation. Now, what is the actual reason for the different kind of allele, different kind of antigen synthesis that we can think here? IA allele is basically encoding enzyme that is called N-acetyl glucosamine transferase. That means this would transfer the N-acetyl glucosamine. Similarly, IB transfer the galactose. Now, where to transfer? These enzymes would add the concerned sugars to a mucopolysaccharide called H structure found on the surface of RBC, and thus they modify accordingly. So, this H structure is fructose, galactose, and acetyl glucosamine. Under the influence of IA, the N-acetyl galactosamine is added, while under the influence of IB, there is addition of galactose. So such kind of modifications can be done. The small I allele, that is not specifying any kind of active enzyme, that's why the H structure remains the same. In the better diagram, we can see here, this is the H structure, H substrate, IA allele, where the N-acetyl galactosamine is basically transferred here. Similarly, IB basically transfer the galactose. So this is the biochemical genetical studies of ABO blood type. Thank you so much. Stay blessed.